Hi guys. Today's lecture is going to talk about eukaryotic gene expression. Hopefully you watched the lecture on prokaryotic gene expression, which talked about operons. In eukaryotic gene expression, we don't have operon control, but there are a lot of different things that control what proteins are made and what genes are expressed, and some that are turned on and some that are not, okay? And so keep in mind a couple of things. Only about 20% of genes are expressed at any one time in a eukaryotic cell. So again, we've talked about this idea of all DNA is the same. Every cell in your body has the exact same copy of DNA. And so your skin cells, the cells that make up your liver, the cells that are making up your neurons, your nervous system cells, your cardiac muscle tissue, all of those cells have the exact same DNA in it. So why aren't they exactly the same cells? And that's because of expression of genes. And as you can see, only 20% of the genes of a DNA strand are actually expressed at any time in a particular type of eukaryotic cell. And so if we think of DNA, a long DNA strand, and we change it into proteins and so forth, and there's so many genes in that DNA, okay? There's thousands upon thousands of genes, and, if we just number them 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, in this case, like let's say that a neuron, okay, a nervous system cell. In a neuron, maybe we need genes 3, 4, 6. Okay, in a muscle cells maybe only read or turn on genes 7 and 8. Okay, and so it's not so much that they don't have those genes. I mean, a neuron contains all genes 1 through 10, but maybe only genes 2, 4, and 6 are actually expressed, okay, or read in order to make proteins. And so that's why a neuron has a different job, different function, and a different structure than what a muscle cell would. And keep in mind, remember, it's the structure that determines its function, or function that determines structure. Those two things go hand in hand. And so if I structure the neuron to only have genes 2, 4, and 6, its function is going to be different than the muscle. Okay, muscle cell. So do keep that in mind when we're talking about this. What we call this is differential gene expression. And so we differentiate the genes that we need to have turned on based on the function that we want that cell to have. Again, it's not that they don't have them. They do have those genes because the DNA is exact copies of each other. Every cell has the exact same DNA, but not every cell needs all of those genes expressed. And imagine the amount of energy and the amount of transcription and translation and the amount of enzymes that would be needed to read every single gene on each strand of DNA. It's just not worth it. It's not worth the time. It's not worth the effort. It's not worth the energy. If every cell had to read all of them, but only use so many of them for their function, it just doesn't make sense. So we only read so many genes around this, okay? And so based on that idea, there are a couple things that can enhance how many genes are transcribed, or can also take back from the amount of gene expression that you have. And so eukaryotic gene expression can be regulated in different ways. And the first thing we need to realize is we need to talk about a couple of things that maybe you've seen, maybe you haven't. But there's something that we call a histone. And a histone, I like to think of it as kind of this ball of Velcro. Okay, it has these little kind of arms that stick out of it. If you ever seen those little Velcro, or I don't even know if that's what you call them, but they're like these little balls that you can make art out of now, that they look like they're just Velcro and they stick everywhere. My kids have them. They're in the carpet. They're in my hair all the time. But anyway, that's what these things look like. Okay, they're histones. And a histone is going to be what DNA wraps around. And so DNA will wrap around these histone proteins. A histone is a protein. And the DNA will wrap around that protein, and it'll call, and the histone DNA complex is going to be called a nucleosome. And a nucleosome, these histones, okay, with the DNA wrapped around them, what'll happen is that you'll get a bunch of them. And I said they kind of are like those, you know, sticky Velcro things. When the DNA is wrapped, histones will interact with each other and stick together, and that DNA will come very tightly packed together, okay? And so this is how we take our chromatin, which, remember when I talked about chromatin before, we just say that's kind of like what DNA looks like before it turns into chromosomes. So during the G1S G2 phase of the cell cycle. And in the mitosis phase, remember, we take that chromatin and we condense it into those nice tightly coiled chromosomes. This is what we mean. This is how they coil. They use these histones, these nucleosome complexes, in order to stick together to tightly coil into those chromosome features. Now, with that being said, the histones sometimes can be more tightly packed or less tightly packed, and depending on if they are very sticky or not as sticky, that will mean more or less transcription, which means more or less protein being made, okay, which means more or less gene expression. And so there's two things that kind of affect this. And so DNA can be methylated, and we call this DNA methylation. And a methylation is where we add a methyl group, and you don't need to know what a methyl group is. It's a CH3, but don't worry about that. So we add a methyl group, okay, onto this nucleosome, basically onto the histone. And what that does is the methyl group, it makes the histones bind together even more, okay? So it's gonna make these things even packed tighter together. And so with that methylation, they almost act like extra sticky groups, okay, to pull those histones in tighter. Well, if I make my histones tighter, if this is my DNA and I make it tighter, keep in mind, remember when we transcribe the DNA, we have to open it up. We have that, you know, helicase that unwinds it and all that kind of stuff. Those enzymes that go through transcription, they're not going to be able to bind if that DNA is extremely tightly wound, okay? That helicase that unwinds the DNA isn't going to be able to unwind it if it's so tightly packed. And so if we add these methyl groups on and we keep these histones together even tighter, what this means is that the transcription, okay, or the making of the protein, the transcription will decrease. Okay, it will go down. We won't be able to transcribe as much because there just isn't anywhere for us to attach to if it's so tightly bound. And so what that's going to mean, less transcription, less translation, less protein being made, okay, which means less expression of the protein. And so this idea of methylation of these histones, keeping them more tightly packed, it's going to keep as much gene from being expressed. Okay, so let's say that we just don't need a bunch of protein at that particular time. When we talked about the bacterial, you know, DNA, and we talked about the operons, remember we had reducible or repressible and inducible. Let's just say that we don't need to make as much. Okay, so same thing kind of happens here. We kind of, you know, have a lot of the protein turned off. Now, can we get rid of these methyl groups? Yeah, we can. We can demethyl, okay, or take away methyl groups, okay, in order to make more protein. And there's a lot of diseases out there where we're kind of trying this, and a lot of our genetic trials right now, there's a lot of diseases where genes aren't being expressed, and that's why children are having issues with some of the disorders they're having. And so if we can go in and take out some of these methyl groups and maybe cause more of the protein then to be made, maybe then we can change some of these genetic disorders. And so a lot of genetic counseling is focusing on that right now, trying to figure out how we can make more protein or make the protein more active. And so to do that, we need to make more of it, get rid of some of these methylation groups, okay? Now, if we can decrease it, there's also a way to increase it. And so there's also something called histone acetylation. Okay. And histone acetylation is basically the opposite of what methylation is going to do. And what, again, we have our histones, our complexes, those little sticky balls that have the DNA tightly wrapped around them. What we can do is we can actually add an acetyl group, okay, on the ends of these histones. And when we add an acetyl group, it actually makes them less sticky. Okay, so they will actually, instead of being tightly coiled together, you might see, okay, the DNA here, and then on down the line, you might find another histone. It doesn't want to, because we added that acetyl group, actually kind of acts as a repulsion factor, and it will repel or cause these things, these histone complexes, to actually separate. They won't be as sticky. And so now the DNA isn't as tightly wound. We have these nice little stretches, okay, that are nice and open. It would be very easy for those transcription factors, those helicases, those DNA polymerases to come in and bind to these areas and go ahead and read the DNA. And so what the acetylation does is it actually loosens things. It actually loosens the DNA, okay, so we can have more transcription, which means then that we could have more protein, okay, and more expression of the gene. So more genes may be expressed, more of the genes might be able to be read, and so this is going to give us an increase in expression, okay? And so these modifications, they actually can be passed on to future generations, and they can also be reversed, okay? And so, we talk about things like obesity sometimes. And, usually you can see kind of a family trend in obesity, but we know it's not technically genetic. It's what we call epigenetic, okay? Epigenetic inheritance is a factor that we can control, but there's also a genetic root to it as well. Things like obesity, we can see there's kind of a trend, okay? If you have, larger parents or something like that, and you yourself maybe as well. And so what we see is that there is a little bit of genetic component to that. However, you can change that genetic component by having the proper diet and proper exercise, or if it's genetically based, trying to find the right, expression, or in this case, maybe methylation to not express as much of a particular gene. And so again, trying to look at how we can weigh environmental factors with how they are genetically based. But epigenetics can be changed based on environmental changes usually, okay? The other thing in this chapter, besides the methylation and acetylation, talks about transcription initiation. So why do we start reading a gene? Like, what makes it start? Like, why do we even think about making more protein? Obviously, most likely there has to be a need. But there's going to be some type of a control factor usually. And we call these control factors activators and repressors. And these are very similar to activators and repressors like we saw in the operon. So it's very similar to inducible and repressible. Okay, so an activator is a complex that is going to bind to a specific region on a DNA strand that is going to tell you to start reading the DNA. Okay. Activators can come in different forms. They can be enzymes. They can be proteins that are not enzymes, but they're going to usually bind to a promoter region. Okay. And just so as we saw before, they can bind to a promoter region, which is also known as the Tata box. Again, that's one of my favorite, the Tata box. It can bind there at the Tata box, the promoter region, and it will cause the RNA polymerase to start reading, to bind there and start reading the rest of it. Okay. On the other hand, repressors, this is also a complex. Again, they can be some type of protein complex, some type of enzyme complex. They're going to do the same thing. They are going to bind, and they're going to bind usually at the Tata box, the promoter region, and they are going to cause the protein to not be read. Okay, so let's say we've made enough protein. So again, it's a negative feedback system. Let's say we've made enough of a protein. This repressor would bind to the Tata box and would block the RNA polymerase. And so it would block, RNA polymerase. It would block the RNA polymerase from binding. Okay. On here, it would cause the RNA polymerase up here to bind and then continue to read. And so the activator sounds much like an inducible operon, whereas the repressor sounds like a kind of repressible operon. So do keep that in mind. Okay. Other thing that we can look at, alternative RNA splicing. All right. And so in the last chapter, we talked about this idea of introns and exons. Okay, remember introns are what we call junk DNA. Exons are what we actually use. And so let's say that we have 1 exon, 2 exons, 3 exons, 4 exons. We've already taken out the introns. We've already taken out the junk that we don't need to read. Well, just because we took out the junk and left with exons doesn't mean that we're going to use all those exons. So we might only use, maybe I only need exons 2 and 3. And so I alternately splice together 2 and 3. I get rid of the rest. Okay. And those are the genes that'll be read to, let's say, make my neuron, as I was saying earlier. Well, let's say I don't need 2 and 3. I need 1 and 4. And so I can get rid of 2 and 3 and I can splice together my exons 1 and 4. Instead, that gives me a muscle cell. Okay, this is what we call alternative RNA splicing. All the genes are there. We've transcribed them. But what we're going to do is we're just going to read some of them. Okay, and only some of them. All right, so that is the gist of our eukaryotic gene expression. If you have any extra questions, let me know. I know there's a web quest on epigenetics that you need to complete, and that'll kind of walk you through some of those ideas of how environment plays a role in the genes that you have and how they're expressed. All right. Any questions? Let me know. Email me. I'll talk to you guys next about biotechnology.